All right, so we finally made it to the current DNA profiling process. So we've kind of stepped our way all the way up to this by learning all the different parts, and hopefully we can pull it all together to show you that this is what the FBI currently uses in the CODIS database to run somebody's DNA for a profile and compare it. All right, so step one. The cells are broken down to release the DNA via extraction, and that part is just like we learned, right? Lysis solution is added, the cells are broken down, they're put in a warm water bath, they're put with alcohol, right? The whole process, just like we learned, okay? So step one, the cells are broken down to release the DNA via the extraction process. Step two is similar, but instead of the DNA being cut with restriction enzymes, now what they do is they add a PCR primer, and we talked about the polymerase chain reaction being the reaction that allows for lots of DNA to be copied quickly, and we learned how that process works, and we know that primers have to be added to find the location in the DNA in which we want to copy. Well, in this case, there's a PCR primer that's added, and it's fluorescent. So they add a little fluorescent marker to it, okay, to locate and copy what's called STRs. And we're going to talk about what an STR is in just a second, okay? So PCR primers are added to the DNA to locate and copy what's called an STR, which is a short tandem repeat. And then they actually do PCR on that location in the DNA. STRs are the most used of all the different forensic markers that are out there, and they're the data that gets put into the FBI's CODIS database, which is the database that they store everybody's DNA profiles and use to compare. So CODIS stands for the Combined DNA Index System. Okay. All right, so let's talk about what an STR is, since we kind of just introduced that right here. An STR is called a short tandem repeat. It's 3 to 7 bases in length that repeats itself multiple times in a person's DNA. So for example, this right here is a short tandem repeat, AGCT, right here. And you'll notice in this person, it gets repeated 1, 2, 3, 4, 5, 6 times. So 6 times that repeat, AGCT, was repeated. For person 2, down here, it's the same repeat, AGCT, except this person only repeated it 1, 2, 3, 4, 5 times. And in this person, again, same repeat, AGCT, but this person repeated it 7 times. So the idea is the short tandem repeats that are found in individuals, everybody has the same repeat, AGCT, right? It's just how many times it gets repeated is different for me than it is for you, okay? So what they do is they add a PCR primer to find this location, AGCT. So maybe the primer would be the opposite code for that. So maybe the primer would be TCGA, right? Maybe that would be the primer, and they would put a fluorescent marker on it. So maybe they would make it a different color. All right, and then that primer would go into the DNA, and it would bond, right, because it's an opposite code. It would find this location, and then they would do the polymerase chain reaction on just this section of the DNA, everywhere that this code, AGCT, was, all the way through, and they would copy that location. Okay. This is a little bit more accurate of a representation because you have 2 chromosomes that you get, right? One from mom and one from dad. You have 2 STR repeats for each one. So here is your same repeat right here, AGCT. This is in person 1's chromosome 1. This is also chromosome 1, right? But this is the one that they got from mom. This is the one they got from dad. So person 1 has 4 repeats and then 6 repeats. So they would have 4 and a 6. Person 2, again, same STR sequence, AGCT. This is the chromosome they got from mom. This is the chromosome they got from dad. They got 6 repeats from mom, 7 repeats from dad. And this person down here, they have 5 repeats from mom and 5 repeats from dad. So person 1 and person 2 are what we call heterozygous for this STR, and person 3 is homozygous because they got the same number from both mom and dad. Okay, so let's just do a quick little recap of some vocabulary. This is stuff that you probably already know, but we're just going to recap it in terms of short tandem repeats or STRs. A locus or a loci refers to the location on the chromosome in which the STR is. So each person has 2 copies of every chromosome, right? So you have 2 alleles. So like here's your chromosomes, right? 1 through 23. Okay, you have 1 through 22 and then an X and a Y. Okay, the location or loci refers to the location on the chromosome. So you can see here that this particular STR, which is AGAT, is located at chromosome 5. So it would be on this pair of chromosomes, section 818, which is this location on the chromosome. So the locus is just what chromosome it's on and what location that within the chromosome, so that they know where to like locate it at. And then the alleles refer to the type of DNA, and in this case, your alleles are your number of repeats, how many repeats you have. So for this one, for chromosome 5, this person had 1, 2, 3, 4, 5, 6, 7 alleles, or 7 repeats on this one. And then 1, 2, 3, 4, 5, 6, 7, 8, 9 repeats on this one. So their alleles for this locus would be 7, 9, right? So you know how, like when we talk about alleles for like hair color, you can have a brown allele, a blonde allele, a black allele, a red allele. Well, for STRs, your alleles are the number of repeats you have for that particular STR, short tandem repeat. Okay? The locus is just the location on the chromosome where you're looking for that particular repeat to be at in the DNA sequence. All right, so a DNA profile can be different for each person. So if this was the STR we were looking at right here, okay, the D5S818, which we just said previously, the repeat was AGAT. Okay, so the repeat that we were looking for was AGAT, okay? And we were looking at 5 different people. Here were their different repeats. So Wes repeated AGAT 11 times on one of his chromosomes and 12 times on the other. Erica repeated AGAT 12 times on one of her chromosomes and 13 times on the other. Jeremy repeated AGAT 11 times on both of his chromosomes. Lori repeated 10 and 13 times, and Raina 11 and 12. Okay? So when you're looking at this, not everybody has the same number of repeats. However, we can see that Wes and Raina do. So if we were looking at that and only this one STR, their DNA would look the same. Okay, but we already said that DNA is individual, which means nobody has exactly the same DNA. So let's kind of show you why. The FBI uses 13. They actually bumped it now to 20. But there's 13 common loci used in the CODIS database, and here's what they are. Here's the names of them. Here's what chromosome they're on, and here's the repeats. Like the TPOX STR marker is on chromosome 2, and its repeat is AATG. You can get between 6 and 14 repeats on it, right? And some of these other ones down here you can kind of go through. So here's the 13 core STR loci. They actually started to use 7 more in recent years, but these are like their really big core common ones that they've used in previous years too. And here's the locations on the chromosomes that they're at. The AMEL, A-M-E-L, is the sex determiner, whether you're male or female. That one will tell you that. It's on the male, female. It'll tell you if the person is male or female. And then all the other ones will just be STR repeats. So now let's take a look. Now we have 5 STRs. So up here at the top, we have 1, 2, 3, 4, 5. 5 separate STRs that we're going to take a look at. So we went through and we extracted DNA for Wes, Erica, Jeremy, Lori, and Raina. And then we put in PCR primers to find these 5 locations in their DNA, and we copied just those 5 locations. And then we went through the whole process, and here's what we found, okay? This is Wes. Wes, for the D5 section 818, he had 11 repeats and 12. For the D7 one, he had 10 repeats and 11. For the TPOXX, he had 913. For the D13, he had 1111. And for the VWA, he had 15 and 19. So can you see anybody who matched at all 5 locations? You shouldn't be able to. You can see at this one, Wes and Raina matched, but then at the next one they don't match, the next one they don't match, the next one or the next one. And then you can see that there is, at the TPOXX, Jeremy and Lori matched at one of them, but not at the other ones, right? So it's possible for you to match at some locations, but you're never going to match at all 13 locations, which is why they do so many, right? You're never going to have exactly the same repeats as somebody else in 13 different spots in your DNA, which is why short tandem repeats work so well to compare your DNA with somebody else's, or your DNA with crime scene DNA or something like that. Okay. So now that we know what short tandem repeats are, all right, so step 1 is to extract the DNA. Step 2 is to use the polymerase chain reaction, PCR, to find the STRs in your DNA and just copy those locations. So that's the only part of the DNA we're copying. Now what we're going to do is we're going to take those STR fragments we just copied during PCR, and we're going to put them into what's called a capillary electrophoresis machine. So remember, we learned how electrophoresis works previously. It takes electricity and it moves DNA through a gel, and the smallest fragments go further, the biggest fragments don't, and it separates the DNA according to size. Well, all they did was they were able to figure out how to make this on a small scale and then be able to get a digital printout from it instead of having to use the gels all the time. And so here's how it works, okay? The ends of a bent thin glass tube are immersed in the buffer liquid. Okay, so here's buffer and then here's your tube right here, okay? So this is the thin glass tube, all right? Inside the tube is coated with gel. So the tube is lined with gel all the way through, similar to the gels that they use in electrophoresis. And then the DNA solution is injected in one end with a syringe. So usually it goes in right here with a syringe, okay? So the DNA got added right here, and it's going to run through this tube. And as it runs through the tube, the negative charge makes it move so it goes towards the positive charge, right? And the 13 STR fragments are different lengths, and they move at different speeds, same way as before. The shorter ones, the ones that maybe only have 6 repeats, will go faster than the ones that have 12 repeats, right? And so as it travels through this tube, it goes right here by this detector. And the detector actually then detects what's there, the fluorescence that the PCR primer added to it. It's a laser detector, and they identify it and they record it on a graph. Okay, so they identify the PCR primer and they're able to record it on a graph. And then this is what you get when it comes out, okay? And I'm gonna actually stop this right now, and I'll do a part 2 video because we won't make it much longer.